Good morning. I'm Daryl Brugink, Executive Editor with No-Till Farmer, and I'd like to welcome you to this morning's webinar where we're going to look into the issues with getting no-till crop residue to decompose in a timely manner and the value that no-tillers can gain from timely residue breakdown. Today's No-Till Farmer webinar is sponsored by Biodyne-Midwest. They are the manufacturers of Environoc liquid microbial products, and we do want to thank Biodyne-Midwest for supporting today's webinar and making it possible. We also want to thank our presenter, Dan Coffin. He's a certified professional agronomist from Fort Wayne, Indiana, and he's going to help us understand the biology of crop residue decomposition and the importance to no-tillers. So with that said, I want to thank you again for joining us, and we're going to turn the controls over to Dan Coffin. Good morning, Dan, and thank you for leading today's webinar. How are you doing?
Good morning, Daryl. Yeah, thank you for having me here. I'm doing well. I'm a little tired. Had a long night here with a bunch of high schoolers, but everything's back to normal here, and we're ready to rock and roll and talking about managing residue to make some money. For those of you who are new to this idea, managing residue to make money is the whole idea of taking what you have as an asset on your farm. We're going to be covering that. And we really appreciate you folks taking the time to be with us this morning as we try to run through some ideas that may help you, especially in a year that's going to be rather challenging economically. Just so you have a little bit about my background, because I don't ever really talk to people without helping them know that I have had a lot of time and experience behind this. I grew up on a grain farm in East Central Indiana. Started detasseling corn when I was 11, so I had to bend the corn plants over just to get the tassels out. But as a young boy, grew up around a lot of vegetable production and scouting. My father was a scout for two major pickle and tomato companies, so I was very, very familiar with that. Went on to school to get a bachelor's and master's in crops and soils from Purdue University, where I taught as a TA in the soils and the soil fertility lab. So I have a lot of background in helping people understand how those systems work. I was an AMSI tech rep when I got out of school in '87 and '88 when Scepter and Pursuit first came out up in the Northeast U.S. and spent a couple, 3 years with them. I turned into a technical sales specialist in South Jersey for horticultural crops and agronomic crops. If anything's grown in the United States, it's almost grown in South Jersey, with the exception, obviously, of bananas. And so I had a lot of time working with a lot of different chemicals, pesticides, and fertilizers, and I earned my CPAG there in 1991. I was a regional agronomist for Northrop King Seeds in Indiana and Michigan for 5 years, and then promoted to the Eastern Corn Belt agronomist for Northrop King for a season. I could see that was going to be a train wreck because I was trying to teach people to be agronomic salespeople, and they actually didn't want to be that, believe it or not. So I went to work as a sales agronomist and an area sales manager for Ag Spectrum Company for a very few years, and then started my own private business back in 2001, where I'm the current president and owner of SP&C Corp, and I do a lot of speaking on a national basis as well as doing some consulting. So had a little bit of time to work with those situations so that you folks know I have a little background to talk about this. So the objectives for today are really a few here. Understand the big picture of residue management, not necessarily all of the micromanagement, but the big picture of residue management, so we have an idea and understanding of what's really going on. Understanding some of the physical processes that are going on in the soil that interact with this cycling. We want to familiarize ourselves with what things are hindering the process from working effectively, because in most cases there is something always that can be going wrong, and it's a matter of understanding that to see if we can fix it. Now let's familiarize ourselves with the nutrient cycling capability of the soil and residue interactions that go on. And then we'll discuss a few options that are available today to enhance the degradation process and improve soil quality and integrity all at the same time. All right, so what is the big picture? Well, as a student of crops and soils, we recognize that cycling is something that has always been here and always will be. Unless the world gets completely sterilized, microbes are going to have that impact on it. And there are a lot of microcycles as well as great big macrocycles that are going on simultaneously. We recognize that in agriculture. I mean, my goodness, we're growing animals from basically eggs to complete animals, harvesting them. We see cycling all the time, and it's something that's very common to us. When we look at the residue that's out here, a lot of people call it trash, and they treat it that way. That's a word that really shouldn't be used. We like to use it amongst ourselves because we understand it. But in a world where we're constantly working with other folks, the other 98% of America, they like to look at things like that and and use those words badly. But some people actually treat it like trash. They may burn up residue or let it fly away. Obviously, if you had flooding situations that moved it off-site, we know that a definition of a weed, for instance, is a plant that's growing in the wrong place. Maybe residue that's gotten moved to the wrong place by natural forces might be considered trash, but in most cases it's not. It's residue, and we should be treating it that way. In a proper term, it's going to be residue, fodder, stover, or crop stubble. These are terms that we should be using, especially even amongst ourselves, but definitely when we're interacting with the general public so that they understand what kind of things we're talking about. Now, residue represents a huge opportunity and a huge challenge. Just depends on your point of view, and we'll talk about that here in a minute. But most importantly, we have to look at residue as a valuable asset to our farm, not just something to get rid of. Because in most cases, when we see all that out there, it is kind of frustrating to realize that, oh, we have another year dealing with something that we don't want to deal with. But there are ways to be able to put it to work for us, and we recognize some of those opportunities as we sit around and talk about it amongst ourselves. We have had a lot of changes in farming practices that have changed our approach to residue management. Now, those of you who, I'm 53, those of you who are my age or older, obviously remember even back into the 60s and 70s when many of us were still plowing. We have not been no-tillers forever, but when we were plowing and burying residue to get it away, where we like to see the black soil in Illinois or a lot of the gray soils here in Indiana, we used to turn that under, and oftentimes if we did it too deep and we shut off the oxygen to that, oftentimes we could go back next year if we put another tillage implement in the ground, and we may be able to see some of the stalks that were nice and bright and yellow from a year ago completely, in some cases, still in the same format. They basically were turned into a silage situation, and had a little bit of formaldehyde, and boy, they were perfectly preserved. So our changes in farming practice has changed our approach to residue management. Now, is it a challenge or an opportunity? If we look at the first opportunity that we see is residue in a cold, wet spring on flat ground. Well, is that really an opportunity or is that a challenge? Most of us would say that's a challenge. A cold, wet spring on flat ground, things aren't draining. Here it sets on the surface, keeping the moisture from escaping, keeping the ground insulated and keeping it cold. So germination is difficult as well as even getting planted at all. What happens when we go to that same situation but on a hillside? Well, those of you who know they're in the government program and you have highly erodible soils, you'll find that having residue in a wet spring on a hillside is probably a good idea because it's keeping your soil intact. So really it's all in your point of view. Residue in a hot, dry spring, well, we had some of that this year. We had started off a little wet, but it got very hot and dry. Well, if we can get planted, then residue in a hot, dry spring is actually an opportunity because it helps keep that moisture from evaporating, keeps the soil a little bit cooler, and allows the crop to go ahead and grow through some pretty difficult conditions. How about residue when you're planting? Well, that's the number one complaint we get from everyone, is that hairpinning and all situations where we get way too much residue in the planter, stuffing up the planter, getting down in the furrow, inhibiting germination, making a mess, that is a real challenge. And so by changing that residue over from something that's stiff and wants to hairpin into something manageable is the very important thing to do. The other thing is we know that we need a lot more carbon in our system. However, to get carbon to break down, it takes a lot of nitrogen. And so most people are trying to overmanage residue with lots of extra nitrogen management to try to get the carbon to nitrogen ratio that's been referred to back in their favor. Whenever you have a grass like the wheat that's pictured here with the combine, grasses have a fairly wide carbon to nitrogen ratio, much more than 20 to 1, maybe 30 to 1, 40 to 1, 50 to 1, 60 to 1. And when you have that much carbon, you have to have a lot of nitrogen to break it down. So things that are above 20 to 1 require lots of nitrogen. Things that are below 20 to 1, like legume crops or some broadleaf crops, especially legume crops that might be 13, 14, 15, 16 to 1, when they start to break down, they'll actually release nitrogen to that soil system. So they're very effective to break down, and that's why you'll see soybean fodder break down faster than corn fodder. And if you had something that was 10 to 1 or lower, it would be very, very quick to break down as well. Now, in many states, especially where we have corn following corn, there's a lot of stover building up. Those of you who are familiar with last year and this year here in the Hoosier State, Indiana. We had an excellent crop last year. The field on the left is last year's crop. Field on the right is some of the yields we're seeing again this year. We've had 2 record crops in a row in most cases in Indiana and parts of Northwest Ohio, and we may have 6 to 8 tons of stover floating around surface at harvest. When you're looking at taking at least 10 to 20 pounds of nitrogen per ton to break down fodder, you're looking at a boatload of nitrogen just required to be able to break that down effectively and efficiently. So it's adding up in a hurry. Well, what is residue cycling? Well, it's a process that's directed by microbial intervention from bacteria, fungi, and actinomycetes. These 3 specific types of organisms, all called microbes, working in conjunction with each other, typically in the best sense of the word, is what is directing. So microbial action is always going on. The microbes eat first at the table of the soil, and plants and everything else gets the rest. So this process goes on naturally and effectively unless you sterilize that soil. Their objective is to change over from the more visible crop pieces that we see to less visible crop pieces and invisible crop pieces, and very simplified and in some cases complex organic compounds as they're breaking down. The cells that are held together in all of these tissues are held together by cellulose and lignin. And many of you have heard that term cellulose. You've heard cellulosic ethanol, the challenge to be breaking down cellulose and lignin. If you look up at corn stover, for instance, here's an example taken from Wikipedia. It's a reference from Biomass and Bioenergy from 2009. And you can see that basically when you have the cellulose, the glucan, xylan, ribonan, mannan, galactan, and lignin, all of those pieces added together add up to, in some cases, 55 to 75% of the stalk. So you're looking at things that are very difficult to break down as making up the greatest majority of that stover. Somewhat resistant to water penetration, and they're waxy on the surface. If you think about the corn, for instance, corn is going to have that waxy surface. All grasses have a waxy surface. And it's going to be difficult when water drops on it to get the water to go down through it. It's going to hit, beat up, and fall off. And so it needs to get down to the surface, obviously, but we want that water to try to push through that stalk if we can. It also represents high energy nutrition if properly managed. And I'm referring to that as energy that's going to go into the soil, that is there for microbial growth, for plant growth, for processes that improve soil tilth. All of those represent high energy if they're properly managed. And most of us are familiar with this if we step back and think. We've heard of composting. We're very familiar with composting. It's done in the garden. In some cases, it's done in large businesses now. But above the soil, that turns residues into a more rich and accommodating product to use. Now, what's interesting is if we know people that will take materials off, compost them, and then put them back on those agricultural soils. And the process does result in some interesting things, but it's never the same, because we want that conversion process to actually take place on the surface of the soil so that the soil is constantly being rejuvenated by those conversions taking place. Well, what are some of the objectives of recycling? Well, the number one thing is to open up the stalks, the leaves, and all those other plant parts that are laying on the surface and allow water and air to get inside to wash some of that nutrition that's cell-bound and get it out to the soil. If you look at these grasses, and you can see this is corn, and corn is a grass, and you recognize the stalks, some of the bright yellow and orange pieces in there. Stover itself is made up of all that material, but the stalks themselves have that waxy coating. And so our objective is to try and get water to wash through the center of those stalks from top to bottom and wash the insides out back to the surface of the soil so that the nutrition will wash out. If that's done properly, we create openings for other microbes and insects to get in and continue to break down the material. That is the big objective. The faster that happens, the faster you get little tiny holes cut in the top of that for water to wash in, the faster you get nutrition out, and more microbes can get in there faster, and little insects can get in there faster to break down the material. You know, little tiny beetles and many other insects, as well as little springtail you'll see here on the left side of the screen, and even small mites, predatory mites that get down in there, they're a part of this whole ecological system. And if we have that working for us effectively, everything is working in sync. It's when we start to take out some of these things with different types of genetics that we start hindering, and we'll talk about that here a little later. Another thing is get it down to a size that's manageable for night crawlers and worms to work with. Those of you who've been no-tilling for a long time are very familiar with the fact that night crawlers can do some very amazing things to stubble and residue. If you've ever seen the earthworm middens, the tiny little hotels, if you will, if you go out on a lot of soils, oftentimes in the spring, here in no-till country, you'll see kind of a bearing soil. The soil is beginning to get barren, and you see these little patches of tiny little hills, and those are called nightcrawler middens or earthworm middens. And if they will actually pull that material around to help keep water from running down in the holes more effectively, keep it moist down in there for them to survive well, and also for a food source. So at night they crawl back up and they'll continue to consume that. And most of us are familiar with the fact that night crawler droppings are very, very rich, high richness with nutrition. And in many cases we've heard, for instance, that the droppings can be as high as 900 parts per million or pounds per acre of potash, for instance. So they are a very, very effective way to cycle material. Now, as that material gets smaller, it feeds more microbes, and the smaller the microbes and the more effective they are, they will turn that material into humus or humic acid. You may have heard referred to that, and that's going to improve the soil tilth because it acts as a moisture container, a nutrient container, and a food source for tiny, tiny microbes. It's a small world even for this guy, and you see the arrow is pointing, if you can see the little spider in the center, it's actually sitting on top of its egg sac, and that's looking at the inside of the leftover corn root at the base of the stalk from last year. Now you think about that spider, it's not eating lots and lots of things that are much bigger than it, and so that spider probably maybe half the size of a penny. So there's a lot of insects that have to be out here that are doing the work. So insects in some cases can be just as important as some of these tiny microbes. Now let's think about some things that are not so obvious when considering about this plant that's going to be cycled, because there are some things in here that actually can help us in the whole big picture of cycling. And the number one thing I think about is the plant and the constituents that are left in it before it falls. The beauty of understanding the whole business of turning residue into money is before the plant ever dies, there are things that you can do to it. And some of our smartest growers have learned that when they're trying to fight micronutrient deficiencies, especially boron, in some cases copper, molybdenum, and several of these other very small amount of materials. Here you have a gentleman holding a bean crop that was in excess of 80 bushels. He foliar fed it 3 times. He fed it with the nutrients that were missing from the soil, because if you think about trying to spread those nutrients as granules, very difficult to get even distribution. However, if you foliar feed that crop, not all that nutrition is consumed by the fruit or the grain itself. It's left in the stalk, and so as the stalk lays down, then now you get a much better distribution of micronutrients. So you can actually pre-program your plants to cycle nutrition better for you. Is that plant supposed to fall over as the stalks would stand up from a root ball, or is it going to be completely harvested? We see places all the time, especially you guys up in the plains, where you're leaving a little more residue standing straight up. And so you're going to take the residue from the root ball and hope it eventually falls over. But all of that stuff sitting above that soil now represents unused nutrition that's going to take a while to get to the soil, and then once it does get to the soil, to be cycled further. Or if it's completely harvested all the way down to the ground and the material is spread evenly over the surface, you're going to get a little bit different breakdown curve. What about the size and condition of that stalk before it falls? Because that's going to obviously be considered in how fast, how long it's going to take for that material to break down. And if the condition is poor, if it's loaded with anthracnose, diplodia, gibberella, fusarium, you recognize those names as stalk rot diseases, then you're going to be using the stalk rot diseases to break that crop down further, and you're going to continue to build disease in. Is it nice and clean from the inside out, or has it cannibalized itself and still has this, or does it still have that hemicellulose, that little styrofoam-looking material inside? All of those things are going to add to or reduce the time it takes to recycle. Of course, the more stuff that's inside, the longer it will take. Does the stalk have a lot of openings in it, or does it contain only a few? So for those of you who are using refuge in a bag, and you have conventional hybrids planted out there in and amongst the GMO stuff, if the corn borers are getting in it, like it used to be for us all the time, everywhere when we had corn borer, the more holes that are in it, the more moisture and air can get in, and of course, in the air, there are microbes floating around, bacteria and fungi. So if there are a lot of holes, it's going to be fast to break down. If not, it's going to be fairly slow. Then once it's on the soil, what are the environmental conditions affecting that residue? What's the overall picture? Is it staying nice and warm? Is it staying moist? And so, you know, if it's an early fall and you've got things laid down early and those environmental conditions are perfect, fine, you're going to get a great breakdown curve. If not, it's going to be more challenging. What things are directly influencing the breakdown of stalks? Well, in most cases, it's the presence of the good and bad microbes, and we said by bad microbes, we're talking about disease organisms that are working out there in conjunction with our good guy organisms, and how much of that is there with fungus and bacteria. And so really, what kind of things have been controlling that whole scenario to give you a much better ecological condition, ecology being everything working together in that system? How much nitrogen or phosphate is present in the soil and or the fodder? If you have lots of leftover nitrogen, if you had an excessive season of nitrogen buildup in the crop, and a little bit of phosphate, because most people don't recognize that microbes need phosphate, just like plants need a little bit of phosphate, to maximize their energy. And so oftentimes measuring how much phosphate is left in that crop is going to tell you whether or not you're breaking down effectively. If there's none left, it's going to help slow that system. Are there good populations of insects and worms present to help complete the process? Because even though the microbes do a lot of the work, when it first starts out, a lot of the breakdown process comes from insects and worms. And so if we're using a lot of GMO crops that still have some BT left in them, we're finding out that insect pressure, good insect pressure, has fallen too as a result of having a lot of those BT stalks. Is the soil getting onto the fodder easily, or does it have to be splashed up onto the material? You know, one of the things about that soil is it's carrying, with each and every grain of soil, there are microbes, and so the more quickly the soil gets up on the crop, the faster it will degrade that material, because it's actually using it as an inoculum to carry those microbes. Are frequent fungicides being sprayed on the soil surface? Because fungicides in most cases are non-selective, so thereby they are going to take out a broad range of fungi, and it's going to take some of our good guys out as well as some of our bad guys. And so if you're spraying them onto the soil, that can be a challenge. What things hinder the breakdown of the fodder? Well, obviously your harvest window. If you're down in the south, you're getting out fairly early. You have a long season to be able to break down your fodder. If you're up in the north, like this year, a lot of corn is going to be harvested probably in late October at the earliest, mid to late November. And so if it's already cool and the soil is cold when you start harvesting, then your harvest window is going to obviously be much, much shortened. And so you're going to need soil temperatures probably at or above 40 degrees to really keep breakdown of fodder going. So if it's obviously too cool at that point, you're going to have a lot of material left for next year. What about your harvester equipment? You know, the newer equipment has stalk stompers. One of the new companies coming out with a stalk devastator. Those materials that actually knock down your stalks, crush the tops, break them over, open them up so that everything actually can break down faster. Once those stalks have been shredded at the top, so that they're very open to catching microbes that are flowing in the wind or being splashed up by water, then they're very, very easy to break down at that point. How about tillage, too much or too little? Well, obviously we're talking to no-tillers, and so in most cases that would be too little. Of course, most of us aren't doing anything for tillage because that's why we call it no-till. But the obvious disadvantage of tilling too much is you're constantly putting materials in there, burning up organic matter, taking the concentrations of microbes and delineating them down to a smaller number, where in some cases too little tillage would be just the opposite. You're not getting much soil onto stalks or fodder. And so in most cases, no-tillers, if they're willing to do a little bit of vertical tillage on the surface, just from a light incorporation from something like Phoenix harrows or something like that, that would actually allow you to just get some soil up onto the stalks themselves to get the breakdown process, that can be all you need in some cases to inoculate and start that process moving forward. The weather conditions being too hot and dry or too cold and wet, the extremes of those are always going to have an impact on that. The type of genetics, GMO or conventional, and the culture and the quality of the biology in that soil. If you're using GMO corns, you've got products that are being sprayed many, many times with, in some cases, glyphosate or Roundup. Those products have an antibacterial character, where conventional tillage or conventional hybrids are not going to have that character. If you've got a lot of BT, as I said before, the BT being proteins that actually knock back insects, they're going to keep insects from working hard on those stalks early in the season. And so in some cases you have to wait for that BT concentration to fall. And so the culture and quality of the biology comes directly as a result of some of the things you're doing above the soil as well as things you're doing in the soil. What about the use of cover crops? Something that we know is becoming very, very popular today, and for the right reasons, because, you know, as an agronomist, what I try to help people do is understand that we know that soils are designed to feed crops, but very few of us talk about the fact that the crops are designed to feed the soil. They actually give off materials that those microbes use to grow and serve themselves well. They actually live on the exudates, some of the sugars and other things that the plants growing in the soil, even weeds, give off. So the use of cover crops has become a real issue of helping us to break down fodder more efficiently. And so the type of cover that you use based on your nitrogen utilization is going to have all the difference in the world. In the left, you see a picture where they have done some tillage and they've planted some wheat or rye for cover. And so grass covers are going to consume huge quantities of nitrogen fairly quickly. If you're a no-tiller and you have done that same process, you're going to pull a lot of nitrogen out of that system fairly quickly to feed that grass crop. So it's probably a good idea to put a little nitrogen back in when you plant the crop. On the other hand, where you have a broadleaf cover, or especially turnips or really radishes that make a lot of nitrates for themselves, it's not quite as critical, so you'll get some nitrogen deposition for those. And of course, you realize when those go to break down, they break down rather quickly. So nitrogen utilization is going to be a very important part of that. And then also, one of the things that people are not familiar with or don't know about, or if they do, they forget about, is the balance of the fertility. Obviously, the nitrogen and the phosphorus that we put into that system in the fall to be able to help the microbes break down the fodder are very important. But very few people understand the use of calcium. Limestone, which gives us calcium carbonate, and then when it reacts with acid in the soil, gives us free calcium. Free calcium is very important because each and every cell wall of those tiny microbes is built on calcium. So to create a very good biological environment, you need to have a fair amount of free calcium available for those microbes to grow. And so in most cases in these Midwestern soils and probably Eastern Plains and most of the South, we need to have somewhere in the vicinity of 60 to 70% base saturation of calcium to have enough free calcium just to keep the ecology working, let alone, you know, grow a good crop. And then also the carbon in a free state. The carbon, of course, that they're working on, and some readily available carbon, like some sugars and some other things for them to eat as they're breaking down the big carbon situation that we have in the fodder. Now, what elements are important to be released by cycling? That's one of the questions that was posed. And really all of them, because obviously there are 18 essential elements for plant growth on the current list that USDA has for us in research work. Here's a quick piece that was taken from Bundy at Wisconsin. They did some work at Wisconsin and at the Indiana Ag Experiment Station from Purdue on 150 bushel per acre corn. So this was the nutrient content of the corn grain and stover, and I just chose to put the stover in here for our work today. But this is pounds per acre in the stover, and it's 150 bushels. And I'd say probably in Wisconsin, sometimes the stalks are smaller than we have in Indiana. But you're looking at 51 pounds of nitrogen, 14 pounds of P2O5, and 150 of K2O. You start looking at those numbers, that's nothing to sneeze at 150 bushels, and many of us are going to have 250 bushels, and several places are going to have 300. So when you look at those numbers and you think what you might actually put in and year out, the numbers begin to become astonishing at what's actually in that fodder. And you look on down with the rest of those elements there, calcium, magnesium, and sulfur, zinc, boron, manganese, iron, and copper. These are all things that have been measured, as well as the other few that are still much smaller quantity. When you realize what this is, we're finding out as people begin to understand. Those of you who've been looking at your soil tests for a number of years realize they aren't changing that much. If you're actually turning your fodder over the nutrition in the fodder is doing a lot of the work of feeding that crop in the following season. And that's why if you're putting out lots and lots of P and K, you're not seeing the soil test levels change. Oftentimes, if you're cycling it in the stover, you don't need to keep putting on quantity after quantity. And the big focus is obviously on N, P, and K, because that's the ones that we're buying and paying tremendous dollars for. The carbon is something that we know we need and that we're trying to actually hold in check. From those of you who are very familiar with people talking about climate change and getting carbon sequestered in the soil, all of those pieces of the puzzle are a part of the nutrients that we need to be cycling. And the big cycle of carbon cycling, carbon dioxide in the air, comes from growing better plants and putting that material back in the soil. So also the elements applied in the fall should be focused on helping the stalks break down, not hindering it. The more salted fertilizer you put down in the fall, you drive the salt concentration up in the top inch or two. And when you do that, obviously when you think about if someone was to pour salt on you, that's a dehydrating process. So that slows microbiological development down. So things like huge quantities of nitrogen, phosphorus, and potassium should be avoided in the fall if you can. And things like humic acids, fulvic acids, sugars, things like that with a little bit of nitrogen and phosphorus that help the materials break down actually tend to release then huge quantities of nutrition for you in the spring. So the irony is we either buy it one time and pay for it, or we build it and break it down and get it to be usable for us. If you're looking at a big tonnage, a high fodder year like we are here in the Eastern Corn Belt, that means there's much more nutrition stored in that fodder, as we looked at the numbers before, and much more can be released. So you're looking at the bigger the fodder year, the more nutrition you have available for the following year if you get it broke down effectively. I also make a point here to be cautious in the future with the opportunity to remove the tonnage for cellulosic ethanol. It's fascinating how we as agronomists can take these numbers and look at all of them and say, well, you know, it's not that big a deal. We take off the cellulose and we make ethanol out of it, and it's really not taking off that much nutrition. And if you look at the long-term studies that are being done, they say that there's no significant impact on the nutrition that's being lost. However, the long-term trend is down, and I would say, duh, at that point. You know, it's got to be obvious that we're taking off quite a bit of nutrition one way or the other, and it's actually being pulled from the long-term storage of the soil, from the fact that it's not there and being recycled and building up in the soil. And so as a result, I think that's why we're seeing it fall slower. But I think if we ran that for 5 to 10 years, we'd find that would change in a very few short years after those first 3 or 4. Now, the soil biological activity necessary for effective residue decomposition, well, obviously bacteria. When you look at things like nitrogen manufacturing by bacteria, like Azotobacter, Azospirillum, those types of organisms, Pseudomonas that breaks down phosphate. A lot of that stuff has been understood for a number of years, and we're getting far, far better at it. So bacteria is a very important part. The fungi and the actinomycetes and the mycorrhiza that you may have heard of, well, they develop kind of like what we call a hyphal or a finger system. You see the nice little tentacles of white material growing out from them. They are the ones that can do a lot of work quickly, and they're very, very good with enzymes that they have of breaking down the cellulose and a lot of different cell types. So between the two of them, the 3 or 4 of them actually, they're very, very effective. And the beauty of this is, it used to be when the microbiologist said, when we actually find groups that are socially compatible, that's the day that we'll start to see the big jump. And that's the point that we're at today. When we understand that we can get bacteria to give off waste products and fungi that give off waste products, and they use each other's waste products to build a better community because they're socially compatible, now we see things change dramatically. And so that's where we are in today's world. Throw in the worms and the insects we talked about a while ago, then that biology, along with this whole system now, is working in sync, because the worms and insects give off things that the microbes want, and in some cases vice versa. The soil pH, again, we talked about calcium a while ago. Most people are not familiar with the fact that if you start working at one range of the pH scale or the other, if you're at a high pH up in the mid to upper 6s or in the 7s, you're growing mostly bacteria. The fungi can work in some cases in a limited aspect, but very limited at high pH. If you're at a low pH in the 5s or in the or high 4s, for instance, now you're growing all fungi, and very few bacteria really love that condition. They can still grow, but they don't like it very well. And so it's oftentimes we find the guys that are using a lot of fungicides, when they go out and do their homework and do their soil testing, they find out, wow, I haven't limed for a number of years and my pH is getting way too low. So we put on a little limestone to bring that pH back up into the low 6s, where somewhere, if you had a perfect balance of nutrition, it would be somewhere around 6.3. And lo and behold, you put a little bit of lime out, and bingo, your fodder starts to break down rather rapidly, because you've got the whole spectrum now working back on your crop. How about pesticidal use and its impacts? Well, if you recognize that several of the herbicides we have out here today have antibacterial, actually patents for them in some cases, the more stuff we spray to the soil, if they're antibacterial, then they're taking out some of our bacteria. Obviously, with the fungicides we're applying, we're antifungal, and if we're using a lot of insecticides, especially applied to the soil, late in the season, not early. This is a little different than if you're putting out insecticides with your boxes on your planter for your corn crop. We're talking about mid to late season insecticides that are getting down to the soil. If it's on the crop, it's not quite as bad. If the crop has the ability to break them down and chew them up before they get there, that helps. But keeping that to a minimum directly on the soil is a smart idea. So what products are really available out here today to break assist in residue and break it down? Well, obviously our old standby is nitrogen. Many of us for years have used nitrogen. When I went back to one of my master's degree professors and told him that we were working with biologicals, he says, "Why are you messing around with that stuff? Why don't you just put out some nitrogen?" That's been the old key, because they knew that if we had enough nitrogen, then we could take native organisms and put them to work. And so urea 28-32% has been used for a long, long time and still has effective use, and is a great product, especially when you're looking at total cost. However, in some cases now the nitrogen is going up and it's getting more expensive in some cases to use that over the biological products. Nitrogen with a little phosphate, 1034O, for instance, has been used as well, because, believe it or not, a little phosphate along with that nitrogen makes a big difference. So if you're a guy that's been using maybe 5 gallons of straight nitrogen over the past, it'd be smart to switch to 4 gallons with a gallon of 1034O just to get a little phosphate to give those microbes some energy, and it will actually boost them. If you're really smart, you would take 3 gallons of those nitrogen materials, a gallon of 1034O, and a gallon of ammonium sulfate, because the sulfur helps those microbes make their proteins. Those are little livestock, just like the cattle and us. They need to be able to break down carbohydrates or sugars, but they need protein to do that effectively. So sulfur is a limiting factor in most cases. About 80 to 90% of the soils that I see in the country are grossly deficient in sulfur. Your humic and fulvic acids and sugars are out there. Sugars, we've seen that with kids for years. You give them a Pepsi and boom, they're off to the races, or they run around, they go like crazy, and then they fall asleep. And they wake up and they want to do it again. Well, sugars are the fast burn for anything. Microbes, insects, plants. And then fulvic acids are your mid-range carbons that will actually give you a very nice boost, and many of the products that are coming today are specialized fulvic acids. And then your humic acids are very long chain and will actually feed for a long period of time. Amino acids, which are basically just sugars with nitrogen groups married on them, and enzyme products. Enzymes are basically just many proteins married together to do specific things, and they actually turn on microbes in the soil. They're like trigger mechanisms. They're called biostimulants. We've used those for years, and they're effective. We've had specifically designed stubble digesting biostimulants coming out in the last few years that might have 1 or 2 or 3 or 4 organisms in them, and they are working a little better clip than some of those have been in the past, and they've been included with some of the earlier technologies we talked about. And now today we have new technology that incorporates more recently identified organisms and improved food systems, like for instance those specific fulvic acids. We have companies coming to us with specifically designed food sources to go in there and make them more complete. And as a result, that's where we are today. And so for instance at Biodyne, we have a product called BD1 Stubble Digester. It's a live microbial culture. It has living microbes in it, 25 different strains of microbes that are present in that culture that incorporates both bacteria and fungi, as well as the actinomycetes we talked about before. They have a food source consisting of humics and fulvics with some special sugars in there to be able to light that system up when it's laying on the surface of the fodder before it can actually ramp up and get to the fodder. This will give it a nice quick burns, almost like lighting a match on a fire and getting it started. This will give it a nice, quick, available food source for it to work on that waxy surface of grasses, for instance, break off the wax, open it up, and start to go into many, many millions of tiny little drills to drill holes into those stalks. It's a liquid solution, so it could be combined with nutrients you might be putting out in the fall or pesticidal applications that you might be putting out in the fall. If you're making those combinations, it can be a ride-along trip. It's a rate of about a quart per acre, and it's going to run somewhere in the vicinity of $8 to $10. Now, one of the things that's I think important about this is includes cellulose and lignin-degrading microbes. The greatest majority of these, if it's only 50%, is lignin and cellulose-degrading microbes. And a GMO corn stover trial that we just finished here last summer with native organisms, which you think about the disease organisms we talked about a while ago, as well as some of the native organisms, compared to the beneficial microbes that were in 501. When they actually put that in there, you can see the color on the left. When you have native organisms, you're getting a lot of rot, fungus that's actually doing a lot of breaking down of the stalks and building disease. On the right, you see a nice white, what we call the mycelium, or the little hyphae out there. You got a 69% more organic matter loss in digestion in the same time period when you used beneficial microbes that had those lignos, or cellulose and lignin degrading enzymes in them. So a very, very effective material. The microbes need to be present in high concentrations, and they are in ours. It's one of our proud pieces of our technology, have extremely high concentrations. In most cases, we have in some cases in a capful what a lot of people have in a full gallon. In any given gallon, you have trillions and trillions and trillions of microbes that start a very quick microbial bloom, and it maintains a high breakdown curve for an extended period of time. They're 100% naturally occurring, non-pathogenic, and not genetically modified in any way. We don't want any foreign DNA out here that these microbes can't recognize. That will keep everything working very effectively and quickly and gets the job done for us. Also, decreased damage to equipment rubber from stiff, stubborn residue. As I was talking to one guy up in the Dakotas, he was saying his tractor tires are only lasting 2 years. Now, they're farming several thousand acres, but because they have to run over residue in those northern environments, and we see it around here all the time, you've seen all the chipping and cracking that's going on with tires that are running on an average for these big four-wheel drive tractors now $4,000 per tire, and only lasting 2 years. So that kind of thing you can get rid of if you make that residue nice and soft, and so when the tires run over it, it just basically smashes. It doesn't resist. Also, decreased pressure from volunteer seeds on the surface in the follow crops. We're seeing a lot of people in the last year that started using our products that are telling us when they sprayed it over, and one gentleman that called me this morning, Hal Rogers from Wilshire, Ohio. He said, basically his volunteer seed was so low, and many of the other people said the same thing. They did not have to treat for volunteer corn and soybeans this year. And he said in one field he had somewhere in the vicinity of 30 bushels of grain laying down on the ground on the cobs because it didn't go well through the combine. They were small ears. And he said, ironically, it was degrading most of the materials on the cobs, and he did not have seed coming from that. So if you could just get that accomplished, you'd be ahead of the game. These whole ideas are basically providing good return on investment and increased profits. Guys, if it's not economical, don't do it. And what I've seen is when we're looking at $8 to $10 an acre to do some of these things, it's not a lot to spend with all the return on investment that we get from all the benefits and features that we've talked about before. It's a good idea to use these things just to help the soil as well as help breaking down and getting the nutrition back. All these things added together really make for a good return on investment. Okay, how do you find us? Well, my name is Dan Coffin. You've been listening to me for the last 45 minutes, and Gilman Farley is the president here at Biodyne-Midwest. Here's our address. Here's our emails and basically our phone number should you want to find us, and here's our web address. I am the owner of Soil and Plant Nutritional Concepts, and I serve as the vice president here at Biodyne in agriculture. So if you need to get a hold of us, there's some quick and easy ways to find us. And so that puts to sleep another year of fodder buildup and hopefully a good system of management to hopefully you understand it a little bit clearer. There's a lot more science if you want to go into it, but at this point I'm going to end here and turn it back over to Daryl for questions. Hopefully that's Daryl, you're still listening, I hope, out there.
You bet. We're here. Thanks for presenting that. We do have a few questions here. I want to kick back to you, and we definitely got about 15 minutes here to do that. So if you have any questions, feel free to enter those through the webinar. Type those in, and we'll get to as many of these as we can before the hour comes up. One question here, and perhaps to clarify too, in case we heard this wrong, but fall applied 28%, as you mentioned, is used to help break down corn fodder. The listener here thought that in your slides you also talked about salt hindering the breakdown process. So can you talk about, when you're using applied nitrogen in this case and salt, can you also have some conflicts here with trying to break down using nitrogen?
Typically, if it depends on your quantity, Daryl. I mean, if you were using maybe 10 or 20 gallons in no water, and you have applied direct salt directly to the surface of the stalks, there would be a high salt concentration, and that would have a little bit of a hindrance. But however, you'd find that over time, that smaller quantity of material is not going to be as much of a hindrance as if you were to put out several hundred pounds of dry fertilizer on the surface, because your dry fertilizer is going to go right to the soil, where a lot of your nitrogen that you're spraying on surface residue is probably going to adhere fairly closely to that residue. And so in those quantities, most people don't use more than about 3 to 5 gallons. On an average, I'd say a lot of people are using 5 gallons of straight 28%, and normally they're putting that with a little water, so that dilutes the salt concentration. So it maybe on the surface I might have made it sound like that, but in most cases that's not going to be a big issue. It's the big quantity of salt where you're putting out 200 to 400, or I've even heard 750 pounds of fertilizer last fall for this year's crop. That's a lot of salt, and that's kind of material I was referring to.
Gotcha. This is kind of a related question. You may have already kind of answered this too, but it's good to go over it. Cole, if you want to just drop 2 down there. Will the salt that is in 28%, can it kill some of the microorganisms in your product when tank mixed? And again, this may go to levels too that you were talking about. So if you do tank mix them, could you have an issue with that?
I would say probably not if you're going to mix it with water. I mean, normally we would ask people to put out somewhere around 10 gallons of total material just to get a good volume and distribution. There's no doubt some salt is always going to be hard on some organisms as they come in with it. But that kind of quantity, when you realize that in most cases we're talking about trying to put out, in some cases, 1 billion or 2 billion of microbes in 20 drops of water, if you have a little bit of loss, it's a small thing to actually worry about, because if you understand microbiology and these good guy organisms, they'll have somewhere in the vicinity of a 5 to 20 minute reproductive cycle. So that means if there's enough food and enough water and enough calcium and other things to build populations, that one microbe becomes 2 in 5 to 20 minutes. In another 5 to 20 minutes, it becomes 4. In another 5 to 20 minutes, it becomes 8. Unhindered, by the end of the day, one microbe will by itself be in the billions with a B by the end of the day. So they are able to overcome a little bit of salt if there's a situation out there, just from their ability to actually grow if there's a food source for them to grow on. So that would probably be one of the littlest worries I would be aware of. If you're really seriously interested in looking into fodder breakdown, do not let the salt concentration of 5 gallons of 28% concern you.
Okay. So how many, here's another question. How many days, if you've got soil temperatures above 40 degrees, how many days will it take, if you're using an application of fertilizer in a microbial product, how many days does it take for it to be really cost effective? How much time do you feel you need?
Well, you're right. I would say probably at a minimum, I would hope to have 1 to 2 weeks of good activity to start to see some good impact. We had some folks last year buy some product from us here in the fall. They probably were able to get that product on at the earliest late October. I know when we sold the product, it was getting close to November. It was sprayed on, and their comment to us was it was amazing how quickly they were treating fields side by side, in parts of the fields. And before the snow cover that we had here in northern Indiana got on, we had comments from some local agronomists that said, wow, you could actually pick it out to the line in the field where things were sprayed with just a week or 2 of good activity. And even if we get some snow as an insulating cover, if the soil is still warm underneath, If the temperatures are above 40 degrees, you're still going to have some activity. Now, obviously above 50 would be better, but there have microbes that'll work, in some cases at 40-degree temperatures. And so if you get an insulating layer, and let's say you had not a deep cold winter like we had last year because we had frozen ground underneath the snow. If you had thawed ground underneath the snow with an insulating layer, it would also be working, some of those organisms would be working slowly but surely away on those stalks, not the whole group of them, because they're born to be, in more warmer conditions. But if you had, a good week to 2 weeks of activity, that's still going to be helpful. I also have the question from time to time, if I want to till this in, for the guys that do some tillage, if they're listening in, I always recommend at least 72 hours, if they can get that, sprayed on the surface and allow it to work, and then lightly incorporate it so that you've got the microbes, you've got the ability to spread those populations, get it nice and evenly over that surface fodder before you till it down a little bit, and that would give you a little bit of help.
Okay. Hopefully I'm going to ask this question the right way. The question was, at what percentage of humic acid will the microbes start to die off? And there's also a related question whether humic acid is food for the microbes. I don't know if does that make sense? Can you make sense of that question?
Not so much in what percentage of the humic acid would they die off, unless for some reason they were worried about the acid itself having an impact on the microbes. Typically, we look at humic acid as a carrier, as a food source for microbes. They love humic acid. They love fulvic acid slightly better because the chains are not quite as long and they can work on those more effectively. But in most cases, you look at the quantity of material that we put out, oftentimes it may be a quart to the acre. There are people who use a half gallon. There are people who use a full gallon of humic acid to be able to be a part of the food source. And in all those concentrations, which is really about all I'd recommend, when you realize how much exchange capacity that humic acid has, you could take a sandy soil, for instance, and start to have a low cation exchange capacity. You start overloading the soil with too much humic acid, you can actually send things backwards because there's a lot of carbon to chew on, and so you need more nitrogen to make that work. So humic acid is a very good product to be able to feed microbes and get them to bloom and blossom. But I'm not familiar, and maybe someone else may be on that, but I'm not familiar with humic acids in most cases having a really negative impact as long as it's held in check and how much you put in the tank.
Okay. You had mentioned that, I think it's not probably only herbicides, but there's some, different pesticides that have antibacterial activity. What would be some of the common ones out there that that folks need to be aware of?
Well, sure. I mean, number one is because our largest concentration of pesticide is basically today glyphosate, Roundup products. Most people may be aware or maybe not aware that product had an antibacterial patent, and it was one of the most underused patents for years. They actually recently just pulled that patent to be able to use it, and they're actually developing Roundup and the glyphosate organism, or excuse me, the glyphosate compound as an antibacterial agent. So it does have some antibacterial effects. So the more often that we're actually spraying it on the surface, it could have some impact on the bacteria. A lot of the work still has to be done on that, and so we don't want to scare people away telling them, look, don't be using glyphosate anymore. That's ridiculous. We're in a glyphosate society. But just realize that the more times that actually that gets sprayed to the surface, that it can have some antibacterial effects. And then when you combine that with the antifungals, you're really putting a lot of pressure on the surface of that soil to have a decreased microbial activity. So the least number of times that you can actually get it down to the soil, the better. So it's actually better to actually let the plants themselves intercept the glyphosate, use it specifically, and then keep it off of the soils directly, because in a lot of cases the glyphosate will attach very strongly to those soil particles and be there for quite some time. So we just encourage people as best they can to try to keep the glyphosate off the soil as much as they can.
Okay. Are there any others out there that folks need to be aware of? Any other herbicides, or is that's obviously a big one in our society today, is glyphosate?
Yeah, that's the big one. I mean, I think probably if there was a lot of work done, they'd find there'd be a few others. But I mean, the greatest percentage, I don't think we've seen too much with the 2,4-D Banvel type products, and they're going to be the up and coming new post and foliar products. By and large, in most cases, they're small quantities, application per acre, small rates, and we're not really aware of a lot of them, but just, as microbiology gets bigger and better, and we're looking at more decimal places below, or excuse me, more zeros below the decimal point, we find out more and more things all the time, and it wouldn't be a surprise if we've learned some of those things. However, always realize nature has the ability to recover. I mean, if we've learned one thing with modern agriculture, we recognize that nature can overcome, in some cases, a lot of things that man does to it. May take a number of years, but it's always going to repair itself.
Okay. I'm going to combine a couple questions here, but what, in regards to a biological product like you have in microbial product, what is typical shelf life of a product like that? And then also, what is a typical that you find as a cost per acre? And what do you kind of see as a typical return on investment? I know that's going to vary, but I don't know if you can give a little look at the shelf life and then also kind of the cost and payback of using a biological product.
Yeah, sure. Shelf life is going to be, when you're looking at live microbials, they tend to what we call log off, or little by little their populations begin to die down in the jug. And in most cases, we would recommend a year. So, for instance, if you were buying a product this fall, if you couldn't get it on this fall, you can actually make applications in the spring. Once you start to get a warm up, you get the break in dormancy, you'll get the opportunity to put it on again in the spring. So if you miss the fall application, you can still get it on the spring. And we talked about before, a week or 2 weeks of good activity. Well, you'll have several weeks at that point in most cases. So I would say if you purchased it in the fall, you didn't get it on the fall, you didn't get it on the spring, you'd still have a pretty decent product in your jug by next fall. Once you're beyond next fall, you're probably going to be looking at putting in another half again as much of new product, mixing old product with new product, because you're going to start to lose the effectiveness of that. Return on investment, the thing that's nice about it, you're looking at a product that's going to run you around $8 to $10 an acre. The return on investment is going to come many ways. Obviously, we talked about with the nutrient cycling. My gosh, it's easy to find $8 or $10 when you look at the price of $650 phosphate. If you put on 200 pounds, that would be somewhere around $32.50 an acre. All you have to do is get enough phosphate to be able to get your plant off to a good start next spring, and you've paid for that return on investment right there. If you start looking at the carbon, the increase in soil tilth from using biologicals that people see in less than 2 years' time, their soil tilth changes dramatically. So your ability to move water, hold nutrition, do a lot of different things for the crop growth. You can put whatever dollar figure you want on that yourself. Looking at the rubber, we said a while ago, if you get a tire that was only lasting 2 years that cost you $4,000, and you take that to 4 years, add that up with the number of acres, and you could rapidly see that it's not going to be hard to make a $50 to $100 return on investment for 8 very, very quickly and very easily. It's not hard to get the return on investment for these products when you realize what the capabilities are, and to get much more quick cycling of carbon into that system to be able to feed the soil and plant. That's really what we're after, because that healthier soil system, you get the ecology working right. It's amazing how the good guys outlive the bad guys, and the bad guys, even though they can have a chance to get at you, they don't work on you nearly as hard when your soil tilth improves. And I have a lot of people that will tell you that today. They don't use nearly the quantity of pesticides they used to, just because they've got a much better environment ecologically.
Okay. Hopefully there's enough information in this question here, but can you reduce your fall inputs of dry applications using microbial product?
I would say definitely. When you look back at what we said on 150 bushel corn, if you've got 14 pounds of P2O5, 150 pounds of K2O, and 50 pounds of nitrogen just in a 150 bushel stover, when you look at those numbers, that's what I said before, I think as people realize if they cycle this fodder more efficiently, they are not putting in the fertilizer they used to. We don't tell people to cut it completely out, especially your nitrogen. You can never cut nitrogen completely out. That would be suicide in today's environment. But for P and K, we have a number of people that have done things with biologicals for a number of years now who have chosen not to put on much P and K. They'll put on starter in the furrow. and especially for corn, and they're doing more and more with soybeans with foliars and corn too. They're managing their nitrogen and their phosphorus and potash entirely different than they used to, and they're cutting dramatically back on dry fertilizer in the fall, to the tune of, some guys saying, well, I might put out 100, 150 pounds because that's as low as I can run the spreader, but man, that's all I'm putting out, because with the biologicals in the system, we're cycling efficiently and effectively, and we're getting that back. So there's no need to keep doubling up on issues that are related to fertility, and we can do it with confidence. And I tell people, if you need to do it this year, I've done this for years with folks, Daryl, you can do it 1 year without dry P and K if your levels are even close to being in the medium range or higher. You can do it for 1 year with other systems and manage it very effectively if you have someone who can help you. And I am not worried in the least, because I have people that have done it for 1 to 5 and are still doing it, in some cases 15 years later. And I don't recommend that, because I'm still a soil scientist, but with some input and some help from people, you can do that very effectively.
Okay. I should have gone back to this one question, because it goes back to your comments about overwintering it. If you don't get it out in the fall and you got to take it back to the spring. But what are some of the storage requirements for microbial? And I imagine you got to worry about temperature a little bit with how you're storing it.
Oh yeah, it's always the same thing. Never into freezing temperatures. If you can, keep them at 50 degrees or higher, and don't let them go over 120, because once you get above 120, you're going to start to kill off the majority of those. You don't kill them all, but you'll kill a majority of them off. So, typically in a nice, fairly warm, it's cool in the summer, warm in the winter type situations, and dry conditions. And if you keep the lids tightly closed on them, if you happen to open them, that's fine. Use them quickly, put the lids back on, make sure they're tight, keep them, in a out of the sunlight, direct sunlight, and somewhere between 50 and typically between 50 and 90 degrees, you're probably going to be fine. And if you can keep it below 90 degrees, which in the summer in Indiana and most of the Midwest, 90 degrees is a reality. But as long as it's out of the direct sunlight and less than 120, you should be fine.
Okay, one last question here. This one is, how much soil on the residue does it take to really inoculate the residue with soil biological activity for breakdown, and how much soil moisture do you feel is required for the microbes to function?
That could be a double-edged sword. But basically, just wind blowing over bare soil and blowing bare soil up over the fodder is all that's really necessary to inoculate in most cases. You don't have to cover it completely with soil, just have to get microbes up onto the top of the fodder, and so even blowing soil will accomplish that for you. What was the last half of that question?
About soil moisture. How much soil?
Yeah, the soil moisture. Yeah, soil moisture. If you have dew on the surface of the soil and on the surface of the residue, that's enough for the microbes to work. Just free moisture. For extended period of breakdown down deep in the soil, you're going to need a light amount of moisture, and that's all. Just enough for a light film to be present in that soil system, so that the microbes can move around and do the work. They're not going to move, they move probably micro inches. They don't work over in terms of inches until they're exploding populations, but themselves, they don't move over micro inches probably in most of their lifetime. So just free moisture on the surface of the residue, and then probably, even if you had 5/100ths or a tenth of an inch of rain on the surface all the time, you would be having plenty of microbial activity.
Okay. Hey, Dan, if you can, I think you had a slide that showed your contact info. If you want to just throw that up there real quick, that'd be great. But we're gonna conclude here, and I want to give you just a heads up that when you log off today, you will receive a questionnaire to complete. It's a very simple, easy one, a small number of questions, and your answers are going to help us determine the value of today's webinar and plan for future webinars. And there's also an opportunity for, if you would like to talk to Dan or contact me, you got further questions, you can ask for him to get in touch with you as well. Second of all, we're going to make today's replay of the webinar available to you via email. It's also going to be able to go up on our website. It'll be up there shortly. That way you can go back and review the entire presentation or parts of it, to hear if what you heard was what you really heard, I guess. And there is a webinar archive section on the site where you can listen to other recently completed webinars as well. And then finally, I think we got through most of the questions, but if there's any that we didn't really get to, we're going to send those off to Dan and give him an opportunity to still answer those, and we'll post them with the webinar replay as soon as they are available. So I want to thank Dan again for leading today's presentation. And I also want to thank Biodyne-Midwest for sponsoring today's webinar. And I also want to give a big thanks to our producer, Cole Vandermouse, for guiding us through all the technology here. We do hope that your crops are hitting the home stretch in good shape, and going to wish you a safe harvest season as that's coming up. And we do look forward to having you join us for our next No-Till Farmer webinar. So I want to give you a goodbye for now. Have yourself a good rest of the week.